So this lecture is about the cardiovascular system, specifically blood pressure, the heart, and also thermoregulation. There are two chapters in the Brooker Biology textbook that I would recommend you have a look at. These are a chapter on the circulatory systems, and there's also a chapter on control of energy balance, metabolic rate, and body temperature. So the chapters are 47 and 46 for the fourth edition of Brooker, but if you have a different version of Brooker Biology, then those textbook chapters will obviously be different. So the learning objectives of this lecture are to know the different components of blood and what's their function. You should know the basic anatomy of the heart and also the vascular system. Have an understanding of the factors that influence the blood pressure and how vascular tone is regulated. And then the last part, I'm not going to cover it in so much detail. There are some slides, and also pointed you towards the textbook. So you should know how heat is transferred in thermoregulation and how the vasculature regulates the heat exchange in the skin. So the function of our cardiovascular system can be summarised in one word, and that one word is transport. So the cardiovascular system is responsible for the transport of all the nutrients that the cells of our bodies require. These nutrients are transported within the blood. So that's one component of the cardiovascular system, and it's this continuous circulation of the blood that enables our bodies to transport these materials to all the different parts of our body. Now, in order for our cardiovascular system to work as a transport network, the blood must be continuously circulated, and this is achieved by two other components: the heart, which is the pump which circulates the blood, and the blood vessels, which are the networks of tubular structures that the blood flows through. So just to summarise our cardiovascular system, what we're going to be looking at are the three main components: the blood, the heart, and the blood vessels. And we have what's referred to as a closed circulatory system. So that means that the blood is always confined within the blood vessels within the tubular network, unless you cut yourself or something. So that's the definition of a closed circulatory system. Our blood is regarded as being a fluid connective tissue. So essentially what that means, or what you can think of it, is you can think of blood as being cells suspended or dissolved in a solution, and this solution also contains other dissolved nutrients, proteins, gases, and other molecules. If you were to take a sample of blood and put it in a centrifugator, what you would see is the blood separates into three distinct layers, as shown in the Eppendorf at the left-hand side of the slide. So these three layers correspond to three of the four main constituents of blood. So the upper layer is what's known as the plasma, and I'll talk about plasma in the next slide. The next layer is the leukocytes, the white blood cells, and then the bottom layer is the erythrocytes, or the red blood cells. Platelets, which are important in enabling our blood to clot, these aren't shown in any of these particular layers, but they would be found in the middle layer between the plasma and the erythrocytes. After centrifugation of the blood, the upper layer is what's known as plasma. So plasma accounts for about 30 to 60% of the total blood volume. It's actually mainly water, so about 95% of the plasma is water. It also contains dissolved organic and inorganic nutrients, so this is where you find the glucose that enters our bloodstream after we've eaten a meal. And it's also where you'll find things like there'll be some dissolved oxygen and a number of different waste products which are produced by the cells during their metabolism, so you'll find those in the plasma as well. The plasma is also where we find the hormones which the endocrine glands produce and circulate throughout our body, so a large number of hormones are found in the plasma. And you also find plasma proteins. Some of these are important for things like blood clotting, so you find things like prothrombin, fibrinogen. So these are important for enabling our blood to clot. You also find different transport proteins and other things like immunoglobulins, which are the antibodies and part of our immune system. So these are all elements that you'll find are components of the plasma. The other important function of our plasma is to maintain the homeostasis of our blood. So things like the ion electrolyte composition of our blood, the pH of our blood. These are all facilitated by or within the plasma. So moving down from our upper layer to the next layer in the centrifugated blood sample, we would find the leukocytes, and I'm not going to go into details of all the different leukocytes in this particular lecture, but they have an important role in defending our body against infection and disease. The erythrocytes are the bottom layer in our Eppendorf tube after centrifugation of the blood. So one of the terms that you have to be aware of with regard to erythrocytes is the hematocrit. And a hematocrit is essentially just the volume of blood that is composed of red blood cells. And typically in a healthy human, that's about 40 to 65%. So 40 to 65% of the blood comprises the red blood cells. The red blood cells are like the leukocytes, are produced in the bone marrow, but there's about 1,000 times more red blood cells than there are white cells in our blood. The main function of the red blood cells is to carry oxygen, and the reason we need the red blood cells is that there's no other way of getting the cells in our body the oxygen that they require. So some oxygen can dissolve in plasma, but there's no way that plasma can carry the oxygen to all the cells that require it. So we need this transport mechanism within the red blood cells. So to enable the red blood cells to carry oxygen, the red blood cells have a molecule within them called hemoglobin, and each blood cell has about 250 million molecules of hemoglobin. So hemoglobin is a molecule which comprises two protein subunits, two alpha and two beta subunits. It also contains a molecule called heme, and you can see from the diagram here the heme molecule with an iron atom at the centre of it, and it's this iron that binds the oxygen molecule. So the heme part of the hemoglobin is what actually binds the oxygen. And each hemoglobin molecule can bind for oxygen molecules, and if you do the maths with 250 million molecules of hemoglobin per cell, you can get an idea of how much oxygen can be carried within a red blood cell here. The capillaries are the site of gas and nutrient exchange within our bodies. So capillaries themselves are the smallest blood vessels we find within the cardiovascular system. They have a diameter of about 10 microns, just like about the same diameter as a red blood cell, and they're composed of one cell. Their walls are composed of one cell, an endothelial cell. So it's one cell layer thick between the blood and the tissues. So the capillaries are then the main site where exchange of gas takes place between the blood and the tissues and the blood, and also where the nutrient exchange occurs in the tissues as well. The bottom layer of our centrifugated blood sample are the platelets, and platelets are actually fragments of cells which are important in the coagulation, the formation of blood clots, and stop us from bleeding to death after we cut ourselves. So that's what the function of the platelets are, and they are the fourth component of the blood. One of the learning objectives for this class is to know the detailed anatomy of the heart. So this is really just something you're going to have to sit down and learn for yourself if you haven't already learned it. There's nothing more to it. So you need to know the upper chambers, the atria. We have a left atria and a right atrium. And then the bottom chambers are the ventricles, the left ventricle, which pumps blood throughout our bodies, and the right ventricle, which sends blood to the lungs for oxygenation. So you should know the main chambers of the heart, as well as the main blood vessels that come in and out of the heart as well. So So if you don't already know the main anatomical structures of the heart, then this slide is a good example of the things you should know by this stage. This is just an animation of the heart to kind of let you see how the heart functions as it's supposed to do. So what you should be seeing here is that the upper chambers, they contract first of all, and then the lower chambers contract after following the contraction of the upper chambers. So that really means that all the chambers of the heart don't contract at the same time. If they did contract all at the same time, that would be bad, because it would mean that the heart wouldn't function the way it's supposed to function. Remember, the way our heart functions is that the upper chambers, they fill with blood, and then at last, just as the heartbeat begins, they contract and then push their blood down into the ventricles, thereby filling the ventricles, and then after a brief delay, the ventricles contract and they pump the blood out of the heart. So if the atria and the ventricles all contracted at the same time, the heart wouldn't function as it's normally expected to do it. So the take-home message here is just remember that the upper chambers, they contract slightly before the lower chambers, and that enables the heart to function effectively as a pump. So the reason our heart is able to contract is it's made up of a type of muscle cell called cardiac muscle cells. So within our body, we have three main types of muscle cells. These are skeletal muscle, which attaches to our skeleton, enables us to produce movements. We have smooth muscle. These are this type of muscle cells which are found in the hollow organs of your body, such as your blood vessels, the airways, the bladder. And then lastly, we have the cardiac muscle. So here we can see a cardiac muscle cell. So this is which has been isolated from an animal. And the kind of neat thing about these cells is that they have what's known as pacemaking activity. So if you isolate them from the heart and put them and study them under, or you want to study them under laboratory conditions, they'll contract like this, as shown in the short video sequence here. They'll contract for many hours quite happily. Cardiac muscle, like all the other muscle cells in our body, they're stimulated to contract by an increase in the calcium concentration. So what you're looking at here is a mouse heart, where it's a genetically modified mouse, where the cardiac muscle cells are expressing a fluorescent protein which is sensitive to changes in the calcium concentration. Essentially what that means is, as the calcium concentration increases, these fluorescent proteins will get brighter. So when the heart contracts, then what you'll notice is that there's from the animations, what you would notice is that the upper chambers, the atria, they get brightest first, because they're going to contract first, and then after they've contracted and following a short delay, you'll see the calcium increase in the lower chambers, the ventricles, and then once the calcium increases in the ventricles, that causes the ventricles to increase. So remember, it's an increase in the intracellular calcium concentration that causes all the different types of muscle in our body to contract. So the electrocardiogram is just a way where clinicians can monitor the electrical activity of the heart, and they can find out if the patient's heart is healthy or they're suffering from any kind of underlying heart disease. So to carry out the electrogram procedure, you can see that all that's required is a few electrodes are placed on the man's chest there. And what this does is it picks up the electrical signal as it passes across the heart. So here's a characteristic or a normal healthy electrocardiogram. I'm not going to go into too much detail about the different components of it at this stage, but you should certainly be able to see that it looks to be quite regular, and this pattern, the pattern seems to replicate itself every few seconds or so. The electrocardiogram, so this is just to give you a bit more description, a brief description of the electrocardiogram. The P wave is the period of the electrocardiogram where the electrical signal is passing across the atrium. So it begins with the P wave, so that's the atrial depolarization. Then you've got the QRS complex, and the QRS complex, this is the period of time when the ventricle is going to depolarize. So what we mean by depolarize, that's when it's just about to contract, so it depolarizes and then it contracts. And then the T wave is when the ventricles repolarize. So cells, electrically excitable cells, they have to depolarize and then repolarize before they can depolarize again. But you can see in the trace above, you can see how that pattern, this characteristic pattern of electrical activity is replicated at a consistent period of time throughout the tracing here, and this would be regarded as someone with a normal electrocardiogram. And as I said previously, the electrocardiograms are a good way for doctors to detect whether or not someone has any kind of underlying heart disease that they have to go about treating. So our cardiovascular system can be broken down into two different circuits, and that's the pulmonary circuit and the systemic circuit. So if we think of the pulmonary circuit first of all, what we mean by that, this is the blood that comes from the right side of the heart, from the right ventricle, that's sent up to the lungs. It passes through the capillaries that surround the alveoli, and then they pick up the oxygen and release the carbon dioxide into the alveoli. So what you then have is the blood returning to the heart. So this is now the oxygenated blood is returned to the left side of the heart. It enters into the left atrium. The atria contract, and then the blood goes down fills up to the left ventricle. Then once the left ventricles contract, that sends the blood out via the aorta, and then that's distributed to all the different organs and tissues of our bodies by the arterial and capillary network. At the capillary level, the blood is going to give up its oxygen to the surrounding tissues, and at the same time it will pick up the carbon dioxide and carry that carbon dioxide back to the right-hand side of the heart, the right atrium, and so the process begins again. So you're to follow a red blood cell as it passes through the lungs back to the left-hand side of the heart, and then it's circulated throughout the body. You'd find it takes about a minute for the red blood cell to circle to complete this circuit. And remember, the other thing, as I said at the beginning of the lecture, we have what's known as a closed circulatory system. So the blood always remains within the blood vessels, whether it's an artery, arteriole, capillary, venule, or vein. It's always within those blood vessels, unless we do something like cut ourselves. So we have this closed circulatory system that comprises the pulmonary and the systemic circulation, as shown in the cartoon or the animation here. The blood is constantly reconditioned so that its composition remains relatively constant. So reconditioning organs would receive more blood than they actually require for their metabolic needs. And some of the most important organs in that regard are the liver and the kidneys. So they get more blood than they require, and indeed the liver has a peculiar supply of blood to it, which I'm not going to go into any detail that you'll hear about later, and that the liver picks up or gets some of its blood from the gastrointestinal tract. The brain is the organ which can least tolerate a disrupted blood supply. So it's extremely important that blood supply to the brain is maintained at all times. So if the blood supply to the brain is cut off for a few seconds, what happens is that someone will pass out, they'll faint. If it's cut off for several minutes, then this can cause irreparable damage to the neurons in the brain. Other organs can tolerate a reduction in their blood supply to them depending on their activities. So things like the gastrointestinal tract. And this is important because it means that things like during exercise there can be a redistribution of the blood to the active muscles away from the digestive tract, where you're not really requiring to digest your food. You're needing to supply the oxygen and nutrients to the actively working muscles at the time if you're doing some exercise or whatever. So the distribution can be altered, but just remember that the supply to the brain pretty much stays constant, and it does require this constant blood supply. Otherwise, you can cause damage to the neurons in the brain. We're interested in blood pressure, and what exactly do we mean by blood pressure? Blood pressure is the force that the blood exerts against the walls of the blood vessels as it passes around your body. So you can see these arrows kind of point outwards. That's the pressure that the blood exerts against the walls of the blood vessels here. I'm not going to get too involved in the physics of pressures and fluid movement, but this pressure, which is analogous to Ohm's law, it kind of gives you a good way of thinking about how blood flow and pressure and resistance are all related to one another. Essentially, what the equation is showing is, so F stands for flow, so that's the blood flow. And there has to be, remember, there has to be blood flowing through our bodies all the time. So the brain has to have blood flowing to it all the time. Otherwise, you'll deprive the neurons of oxygen, and it will cause irreparable damage to the brain. So this is extremely, this flow is extremely important. Blood blood flows in our body because of the pressure gradient. So delta P is just the pressure gradient. And I'll come back and I'll explain to you what we mean by pressure gradient. And what we're saying, or what this equation tells us, that flow is directly proportional to the pressure gradient. So the bigger the pressure gradient, the greater will be the flow. And it's inversely proportional to the resistance. So what we mean by that is if the resistance increases for the same pressure gradient, then flow is going to decrease. So try and remember this equation. We'll come back to it in a few moments' time just to try and explain things. So hopefully it won't make it too complicated for you. But if you think about this equation, it helps you to understand when we're thinking about blood pressure. So one of the terms I introduced in the previous slide there was pressure gradient. Again, it sounds complicated, but it's really quite simple. So pressure gradient is simply the difference in pressure between the beginning and end of the vessel. And in a slide or two's time, I've got a cartoon drawing which will make this a bit clearer for you to understand. So pressure gradient is just the difference in pressure between the beginning and the end of a vessel. The other thing to remember about pressure and flow is that blood flows from an area of high pressure to an area of low pressure. So that's just the way it is. When the heart contracts, it exerts pressure on the blood, which increases the pressure on the blood, and this is really the driving force which forces the flow of blood through the vessel. So it's this mechanical activity of the heart which generates the pressure, which causes our blood to continuously circulate, and that's why it's important that our heart always continues to beat, otherwise we have a problem. This slide shows the relationship of flow to the pressure gradient. So pressure gradient is what I was just talking to you a minute or so ago about. So here's we've got two different vessels with two different pressure gradients in them. So the first one, we've got a pressure at one end of the vessel of 50 millimeters of mercury and a pressure at the other end of 10 millimeters of mercury. So the gradient from the left-hand side to the right-hand side of this blood vessel is 40 millimeters of mercury, so it's just 50 minus 10 it gives you the pressure gradient. So in vessel 1, our pressure gradient is 40 millimeters of mercury. In vessel 2, what we've got at the left-hand side, we've got a pressure of 90 millimeters of mercury versus a pressure gradient of 10 millimeters of mercury at the right-hand side. So again, if you do the maths, the pressure gradient in vessel 2 is 80 millimeters of mercury. So you can see the pressure gradient in vessel 2 is double what it is in vessel 1. So assuming, well, they are, the diameters of these blood vessels are the same. So what it would say to us is that because the pressure gradient in vessel 2 is twice that in what it is in vessel 1, the blood flow in vessel 2 is also going to be 2 times that of what it is in vessel 1. Because remember the equation I showed you a few slides back, flow is directly proportional to the pressure gradient. So the bigger the pressure gradient, the greater the flow you're going to see. In the equation we saw a few slides back, the 3 terms were flow, pressure gradient, and resistance. Resistance is simply what we mean, the definition of resistance. It's a measure of the hindrance or opposition to blood flowing through a blood vessel or any kind of tubular structure, and it's caused by the friction between the blood or the fluid substance in the tube and the wall of the vessel itself. So that is the definition of resistance. It's the hindrance or opposition to fluid moving through a vessel. If the resistance to flow increases, what that then means, so if it increases, it's more difficult for blood to pass through the vessel. Therefore, the flow rate is going to decrease if the pressure gradient is maintained the same. So think about the equation. So we had the blood flow is equal to the pressure gradient delta P divided by the R, which is the resistance. If the resistance is going to increase, so the number at the bottom gets bigger, but the pressure change, the pressure gradient doesn't change. You have a big number, a bigger number going into the same number in the top. What that will give you is that the flow is going to be reduced as a consequence of that. And we'll see in the next slide what are some of the things that can change the resistance of a blood. There are 3 main factors which can affect the resistance of a blood vessel, or resistance to blood flow. The main factors are the viscosity of the blood. So again, if you think about it, if it's a thicker, if the blood is more viscous, then there's going to be a greater resistance. So viscosity is one of the factors which influence the resistance. The length of the blood vessels, and again, for us, we don't have to bother so much about the length of the blood vessels because normally the length of our blood vessels are fairly constant. So that's not really going to be something that changes, or it changes quite something that changes quickly. The factor that does have the greatest influence in the resistance, and the thing that we're going to be focusing most heavily on is the radius of the blood vessel, or the diameter of the blood vessel itself. So this is the critical thing that we're interested in, and how this affects the blood pressure. So it's the radius of the blood vessel, and I'll show you in the next slide what changes in the radius of a blood vessel, what effect they have on blood flow and blood pressure. So I said to you in the previous slide that the radius is an important contributing factor to the resistance, and the way that the radius of a blood vessel relates to the resistance, the resistance is proportional to 1 over the radius to the power of 4. So probably that would be a bit difficult for you to understand. So hopefully this cartoon makes it a little bit easier for you to think about it. So what we've got 2 blood vessels, vessel 1 and vessel 2. And what you can see in vessel 2 is that the radius of this blood vessel is twice what it is in vessel 1. So the radius in vessel 2 is double what it is in vessel 1. So essentially what that means is if you put these numbers into the equation here, so for vessel 2, you would see that the resistance, so 2 to the power of 4, is 16. So the resistance in vessel 2 is 1/16 of the resistance in vessel 1. So that means that if we had the same pressure gradient, then the flow would be 16 times higher in vessel 2 than vessel 1 for the same pressure gradient. So this is really just showing you how the resistance affects flow within a blood vessel for the same pressure gradient here. Here are the different types of blood vessels which make up our cardiovascular system, and what you've heard about in some detail from Dr. Coates previously. For us, we're interested in two main blood vessels, and that's the arteries and the arterioles. They're important in the determination of our blood pressure, so that's what our focus is going to be on. The arteries, which are the blood vessels that carry blood away from the heart to the tissues, and the arterioles, these are just smaller, you can think of them as smaller branches of the arteries, which carry the blood to and within the tissues themselves. The main difference between the arteries and the arterioles that we have to remember is just that arteries, they have quite a thick muscular layer in them. They're also they have a much bigger radius, or a larger diameter compared to the arterioles. So arterioles they have a much smaller radius. They have fewer layers of smooth muscle cells in their medial layer, and these are the blood vessels which offer the greatest resistance to blood flow, as we'll see in a few moments' time. Arteries can be thought of as rapid transit passageways for supplying blood from the heart to the different organs throughout our body. So the key kind of feature that enables the arteries to work in this way, or to function in this way, is they have a large radius. So remember, when they have a large radius, they offer very little resistance to blood flow, and that's really important here. They also, and as we'll see this in a moment's time, they can act as a pressure reservoir. So what that simply means, I'll describe it to you in the next slide. But it's kind of like if you think about how our heart functions. So it's not like a pump, a fluid pump, which is pumping fluid continuously all the time. Our heart kind of works like it has to fill up with blood, then it ejects the blood, then it refills with blood, and then it ejects the blood out of the heart. And that explains why we can feel a pulse in our wrist, because of the way the heart functions. So what happens then when the heart is not pumping, when it's not ejecting blood, these large arteries have an important role to play by acting as this pressure reservoir, and I'll show you what we mean by that in the next slide. The other thing that's important to appreciate about arteries is that they have quite a large amount of connective tissue surrounding the outside walls of them. The connective tissue is mainly collagen fibres or elastin fibres. So the collagen fibres, they provide the strength. So if you think about it, when your heart's contracting, it's ejecting blood at quite a high pressure when it comes directly out of the heart into the aorta. So the aorta has to be able to withstand the pressure of the blood and pushing against the walls of it, and that's the function of the collagen fibres there. And then the elastin fibres, they provide some, as the name suggests, they're kind of elastic, so they provide the elasticity to the heart, and this is what enables it to function as a pressure reservoir that I'll explain to you in the next slide. So this is just a kind of cartoon that hopefully explains to you what we mean by a pressure reservoir. So if you think about it, when the heart's contracting and ejecting the blood into the aorta, what happens is that the blood pushes against the walls of the blood vessels, and it tends to want to expand those blood vessels out. Remember, the arteries, they have the collagen and elastin fibres. So the elastin fibres allow the blood vessel wall to be quite slightly elastic, and that means that these blood vessels, the arterial walls, can expand out a little bit, kind of like as shown in the diagram here. So the arteries, the walls of the arteries have expanded out. What that means is that when the heart is then relaxing and refilling with blood, is that these arteries, the walls of the arteries, can push inwards or move inwards back to their original dimensions, and in doing so, they keep the blood moving in the forward direction towards the capillary. So it's kind of, this is what we mean by the arteries function as a pressure reservoir. They accommodate the blood which has been injected into them. When the heart is refilling with blood, when the heart is relaxing, these arteries can then, the walls of them can move into their normal resting diameter, and in doing so they force the blood to flow on a little bit towards the. If we were to measure our blood pressure through some invasive procedure, and what I mean by invasive procedure is if you were to insert a pressure transducer, something that records pressure into the brachial artery in your arm, what you would notice is that the recording device, it just doesn't measure one single pressure, but the pressures actually seem to increase and decrease. And these increases and decreases correspond to the heart contracting and refilling with blood. And they're given they're named accordingly to the cycle of the heart. So the pressure the highest pressure that's achieved in your arteries is what's known as the systolic pressure. And this is the pressure when blood is actively being ejected by the heart into your blood vessels. This is the systolic pressure. And then the lower one is what's known as the diastolic pressure, and this is the pressure that this is what your blood pressure would drop to at the period during the period when your heart is no longer pumping blood. And you can see that there's a kind of cycle here in the trace at the top. So the blood pressure increases and decreases as the heart contracts, and that produces the systolic pressure. And then as it relaxes and refills with blood, that produces the diastolic. So the diastolic is the lowest pressure that the blood pressure drops to when the heart isn't actively contracting. One of the most important, or the factor that we are going to be most interested in today in relation to what are the things that affect our blood pressure, is this peripheral vascular resistance. And again, that's just a fancy word for saying the diameter or the radius of the blood vessels. And this is something we've been speaking about previously. So if you think back to the equation I showed you a few slides back, we've got the flow is directly proportional to the pressure gradient and inversely proportional to the resistance. And remember, the resistance is related to the 1 over the radius to the power of 4. So that relationship I explained to you a few slides back as well. You should remember that. So it's the main factors that we are going to be considering that affect our blood pressure are the diameter of the blood vessels themselves, the arteries, mainly the arterioles, the smaller blood vessels. And the key thing to remember is that if the resistance increases, so resistance can increase if the blood vessel becomes narrower, so its radius decreases or diameter decreases. If the radius or diameter decreases, then the resistance is going to increase. And once the resistance increases, the only way our body can maintain adequate flow is to increase the pressure gradient there as well. So the pressure gradient has to increase in order to maintain the appropriate levels of flow. Some of the other things that I'm not going to be focusing on today which can affect our blood pressure is the cardiac output. It's a term I'm sure you've heard about before. So it's the amount of blood ejected per minute by the heart. Another thing that affects your blood pressure is the blood volume. So if the blood volume is quite high, then your blood pressure can be high. And the opposite is also true. If the blood volume drops, then your blood pressure will also drop. And the other thing, which is an important factor affecting your blood pressure, and it's mainly to do with the elderly patients, is the vessel elasticity. So remember, we had the blood vessels expanding and then relaxing and pushing the blood forward during the heartbeat. So changes in the vessel elasticity with aging can influence the blood pressure. But for us, we're mainly interested in simply the diameter of the blood vessels here. So it's the blood vessels called arterioles which have the greatest impact on the vascular resistance. Typically, arterioles are blood vessels that they have a diameter of about 100 micrometers, so that's about the diameter of a human hair. And there's a small animation on this slide which shows you a drug being applied to the arteriole, and you can see the radius or the diameter of the arteriole becoming reduced as this vasoconstrictive substance acts on the blood vessel, and then another a vasodilatory substance has been applied to the blood vessel to make it dilate. So you can see quite clearly how much of a change you could get, if you maximally stimulate these blood vessels, and how much their diameter and radius is changing according to the stimulus that's been applied to them. Moving on from the arteries to the arterioles, so I've just been showing you the short movie of the arteriole. So the arterioles are considered to be the major resistance vessels in our cardiovascular system. So it's these blood vessels which are the most important determinants of our blood pressure. They also have this important role in the convert. So remember, when our heart beats, so it ejects blood out, and we can feel it in our pulse. So it's kind of like the pressure is going up and down, and that's why we feel our pulse. When it gets to the capillaries, the blood flows much more smoothly, so it's kind of going at a constant velocity, and it's really the arterioles that kind of smooth out these pulsatile the pulsatile flow that would normally occur if the arterioles weren't doing their jobs. So their job properly. So they convert this kind of the swings in the diastolic to systolic to diastolic pressure. It converts these swings in pressure to something which is a bit more smooth, and it means that the capillaries themselves don't experience increases and decreases in blood pressure that you would see in other blood vessels. So it's the radius of the arterioles which is adjusted, or which determines our blood pressure, and that's really, we have no conscious control over the diameter or radius of arterioles. It's controlled by the autonomic nervous system. So it's under, we don't consciously control the diameter of the blood vessels. The changes in the diameter or radius of the arterioles are important because it serves as a way to distribute cardiac output to different organs dependent on their particular requirements. So I said earlier, if you were doing some exercise, you want more blood flowing to your skeletal muscle. So the way your body does that is it maybe causes vasoconstriction of the blood vessels to your gastrointestinal tract, and vasodilation of the blood vessels going to the skeletal muscle. So that means that you can get more blood flowing to your skeletal muscles compared to your gastrointestinal tract. So it's really that's an important function of the arterioles. So they are important in distributing blood to different organs dependent on the particular organ requirement at that time. And the other key thing is they do help regulate the arterial blood pressure through their activity. The reason blood vessels are able to change their radius or diameter is because the walls of them are the medial layer of them, you'll find the smooth muscle cells. So remember previously I showed you the cardiac muscle cell that you find in your heart. So the smooth muscle cells are the type of cells you find in the hollow organs such as your blood vessel. So here's a typical smooth muscle cell. So it has this kind of classic spindle shape here. And what you can do is you can isolate these smooth muscle cells from the walls of a blood vessel and study them under laboratory conditions. So here we've got a single smooth muscle cell, and I think there is a short video there that what you can do is you can apply a substance to the smooth muscle cell, and it will cause the smooth muscle cell to contract. And you can just imagine all these smooth muscle cells within the wall of the blood vessel being stimulated to contract. If they contract, they change the radius of the blood vessel and increasing the resistance of it, and that will in turn increase the blood pressure. So as I said a moment ago, we have no conscious control over the diameter of the arterioles in our body. So our body just changes the diameter of those things. There's a whole vast range of different control mechanisms, and again, I'm not going to go into them in any particular detail. Certainly some of the things which cause vasoconstriction, so our nervous system, in particular the sympathetic nervous system, and the neurotransmitter that's involved there is noradrenaline. So what happens is the sympathetic nervous system, stimulation of the sympathetic nervous system will release the neurotransmitter noradrenaline. Noradrenaline acts on receptors on the smooth muscle cells, and this in turn causes the smooth muscle cells to contract. So when smooth muscle cells contract, then what happens is you'll get vasoconstriction. There are other things which cause changes or to cause blood vessels to vasoconstrict. So there's a number of substances such as endothelin. Angiotensin 2 is a substance which is circulating throughout your bloodstream. It acts on angiotensin receptors. Endothelin acts on endothelin receptors to cause vasoconstriction. Then other things, so we've got blood vessels also have what's known as myogenic activity. So remember we said the heart has so myogenic activities is activity that resides within the muscle itself. So what you can get with that is the muscle cells, in response to certain physiological stimuli, the muscle cells can in turn become activated, and when they're activated, you get vasoconstriction. The other thing that causes in contrast, things which cause vasodilation, if you get a reduction in the sympathetic stimulation, so a reduction in the activity of the output from the sympathetic nervous system, so there's less noradrenaline being released to act on the smooth muscle cells. That causes the vasodilation of the blood vessel and a decrease in blood pressure. Substances, again, some of the things you've heard about before, so nitric oxide is a substance which is released from the endothelium. That acts on smooth muscle cells to cause the smooth muscle cells to relax, and you get vasodilation. In addition, when there's a fall in oxygen levels, so what happens is if there's a fall in oxygen levels, then you get vasodilation. The pulmonary circulation is a bit different. I'm not going to confuse you by going through the pulmonary circulation, but normally if there's been a fall in oxygen levels within the body, what happens is you get vasodilation, and that really is a kind of protective mechanism that hopefully can improve the flow of blood and oxygen, and turn oxygen to the organs that demand it, so that they don't suffer any kind of long-term injury there. So there's a whole vast of different things which can cause vasoconstriction and vasodilation of the arterioles, and by altering the diameter of these blood vessels, you in turn affect or influence the blood pressure. So I said at the beginning of the lecture, or the beginning of this presentation, I wasn't going to go over thermoregulation with you. There are some slides, so you should know the contents of the slides, and there's quite an interesting, there's a small section in one of the chapters on thermoregulation in the core textbook that I would recommend you read for yourself. So the material, you should know the material as it is examinable, but I'm not going to formally go over it with you today.